Thanks everybody for joining us this evening here in Perth. I know some of you are logging in from other locations, so I hope you've had a good day or will be having a good day. I'm Tim Colmer, the DVCR here at UWA. I'd like to welcome particularly the McNamara family members who have joined us, also the current and past panel members who have been involved in selecting our Keiran McNamara World Heritage PhD Top-Up Scholarship recipients who we'll be hearing from in this webinar. And I'd like to welcome those four excellent researchers who are our recipients who will be telling us this evening about their work. We'll get to see each other in the second part of the webinar when we get to the Q&A part of this, this webinar. The University of Western Australia acknowledges the custodians and traditional owners of the land on which any of its campuses are located. At the main campus, Crawley, where I'm speaking, the university acknowledges the Whadjuk Noongar people as the traditional owners of the land on which it is situated. The Whadjuk Noongar remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs and knowledge. We pay our respects to elders past, present and emerging. I'd like to acknowledge also the vital support of the Keiran McNamara World Heritage PhD Top-Up Scholarships for research in conservation and management in Western Australia, and thank the McNamara family for this generosity and their vision. The scholarships are, on a competitive basis, open to applicants across all 5 WA universities to attract the next generation of outstanding researchers to further knowledge and expertise relevant to the conservation and management of the natural values of World Heritage Areas in Western Australia. These prestigious scholarships continue the legacy of Keiran McNamara. Keiran had a distinguished career, including as the Director General of the Department of then Environment and Conservation and the predecessor Department of Conservation and Land Management from 2001 to 2013. Keiran was highly regarded by his colleagues for his knowledge, for his personal qualities, and for his vision for the conservation of WA's beauty and diversity for generations to come. And a highlight of his career was the listing of World Heritage Areas in Western Australia. As I've mentioned, we'll have some presentations from our researchers and then a question and answer session. Between the presentations and the question and answer session, there'll be a short break so that we can enable the video feeds for everybody to participate together. This webinar is being recorded and a podcast will be made available for those who've missed it. We have 4 great speakers and excellent research projects, and you'll meet each of our 4 speakers very soon. I'd like to start by congratulating Molly Moustaka on being the 2020 recipient. You'll hear from Molly and meet her a bit later. To our first speaker, Emily Lester, I'd like to also congratulate Emily on her submission of her PhD thesis in June. Emily has a Master of Biological Science, Marine Biology from UWA, and commenced her PhD at UWA and Australian Institute of Marine Science in 2016. She was the inaugural recipient of the Keiran McNamara World Heritage PhD Top-Up Scholarship. Emily's PhD research is investigating the influence of reef sharks on the behaviour of mesopredators in a dynamic coral reef seascape. Her research used life-size models of threatening reef sharks and non-threatening, for example, turtles, taxa, as an innovative method to examine predator-prey interactions that are rarely directly observed in nature. Emily, over to you. Thank you.
Great, thank you, Tim. So I guess I'll just share my screen, bear with me. So I'm going to give you a whirlwind tour of my PhD research in 15 minutes. But before I get started, I want to give you some background on predator-prey interactions and why we really want to study how prey change their behaviour in response to predators. So predators exert really strong top-down effects on their prey in two ways. The first is by consuming them, and the second is by having effects on prey's behaviour. Because when predators are around, prey species need to be vigilant to avoid being that predator's next meal. So although these anti-predator behaviours reduce the likelihood that prey species pay the ultimate price of predation and get eaten, these anti-predator behaviours actually incur their own significant ecological cost, because time that's spent avoiding a predator is time that can't be spent on really important behaviours like foraging. So these anti-predator behaviours actually have the ability to alter trophic structuring and energy flows of entire communities. So if you want to look at the role of a predator, but you're only focusing on the consumptive effects of that predator, then you're really only getting half of the story. So in coral reefs, sharks are large-bodied predators that occupy high trophic positions. Now we know a lot about their consumptive roles in these environments, but we don't know whether they have the capacity to induce these anti-predator behaviours in mesopredatory species like snapper, groper, and even rays. And that's because these interactions are actually pretty hard to observe, especially by humans, because to reef fish, we're quite scary. And if we want to observe these interactions in person, our presence is likely going to confound everything. So I wanted to come up with ways to examine and quantify these rarely observed interactions. And one tool that I was particularly interested in is baited remote underwater video systems, which are called RUVs as well. So it looks a little bit like this schematic on the left here. It has two cameras that are set within a frame facing a bait bag. And it's a really common tool used to examine fish communities, and basically throw it over the side of the boat. Here's one descending to the bottom of the seafloor. And because we have two cameras, we can, basically what happens is the bait bag attracts the fish to within the field of view of the RUV. And because we have two cameras, we have stereo vision, so we can record length. But I also think these RUVs are an untapped resource for behavioural studies, especially things responding to the presence of a predator like this tiger shark here. We can see what fish respond to the presence of this shark, but we can also see how they change their behaviour and how long these behavioural effects last for. So I wanted to develop some standardised behavioural metrics that can complement the existing RUV measures we get from data like this, so that we can get an insight into the strength of rarely observed predator-prey interactions. So I'm going to take you through the RUV chapters of my PhD, and I had four main aims for these chapters. The first was to see, well, what reef sharks are doing on coral reefs and where they are. The second was to develop some measures, some metrics to measure behavioural changes in response to sharks. And then I wanted to see if these behavioural metrics change over the course of a long-term time period, so something like 60 minutes, and then shorter time periods, like 5 minutes. So for aim one, we dropped a lot of RUVs. So I dropped some RUVs in the Ningaloo reef here. I don't know if you can see my mouse. And then we were lucky enough to collaborate with DBCA and the UWA Meg Lab. So we've actually got over 1,500 RUV deployments deployed throughout Northwest Australia. So that's really important to contextualise what we're finding in Ningaloo within the wider region. And for the sake of time, I'm just going to take you through the results of the most abundant species, the grey reef shark. And there's quite a lot going on here, but I just want to draw your attention to the fact that we got a higher occurrence of these reef sharks in no-take areas. And this was actually a bit of a surprise because sharks aren't targeted by commercial fishers or recreational fishers in Northwest Australia. So we weren't expecting to find a result of management in these areas. But when we looked into it, we found that we were finding a lot of these sharks in these large no-take areas in Ningaloo. And I think it's because these large no-take areas like Point Clotes at Ningaloo, they just have some fantastic shark habitat. Point Clotes, for example, has this great drop-off. It's also got really strong currents that whip around the point, and they also have higher abundances of prey species that sharks like to feed on. So I think when you think about these results in the context of shark conservation globally, a recent paper came out that found that sharks are functionally redundant from about 20% of the world's reefs. So places like Northwest Australia are really important bright spots for conservation. And if you want to conserve these shark populations against future threats, having very large no-take areas that encompass favourable shark habitat is a very good place to start. So that's what the sharks are doing. How can we measure how mesopredators are changing their behaviour in response to sharks? Well, I came up with 3 behavioural metrics. The first was time to arrival. So that's the time that we see the first individual of a species in the field of view of the bruv. And you would expect fish to be more wary and less inclined to approach the bait bag in the presence of sharks. So more sharks, you'd expect a larger time to arrival. We also looked at whether fish fed from the bait bag or not, so that's likelihood to feed. And so feeding is actually a very interesting behaviour to look like, because in the presence of predators, animals tend to forego foraging opportunities to exercise caution and vigilance against that predator. So you'd expect sharks, mesopredators to not feed as much in the presence of sharks. And finally, we looked at delay to feed. So that's the difference in the time of arrival and the time to first feed, which is another good way to measure vigilance. And you'd expect fish to have a larger delay to feed in the presence of sharks because they'd be exercising caution. So did these metrics change in the presence of sharks for mesopredators? Well, to look at this over long time periods, I looked at these behaviours on bruvs where sharks were either present or they were absent. And I looked at the behaviours of 4 different categories of mesopredators. So we've got large, medium, small, and rays. And so when we look at time to arrival and likelihood to feed, there is a lot going on here, but all you need to know is that the presence of sharks didn't actually influence these metrics, but the level of competition, so that's the amount of mesopredators in the field of view, actually had a really strong effect on both of these metrics. We saw that in the presence of more competitors, mesopredators were faster to arrive and they're also more likely to feed from the bruvs. So this isn't quite what we were expecting, but I think it's pretty cool because if you think about it, in order to survive and thrive on a coral reef, you not only have to outcompete, avoid predators, but you have to outcompete your competitors too to gain access to resources. So perhaps over these longer timescales, competition is a really powerful ecological force that I hadn't thought of before. And when we look at delay to feed, things got actually really quite interesting. So again, we found this effect of competition, but it looked like there was some interspecific competition going on, especially for our small mesopredators. So in the presence of lots of small competitors, so things the same size as them, there was less of a delay to feed, which means they were just trying to take advantage of the resources. But in the presence of larger competitors, so these large mesopredators, there was actually quite a big delay to feed. And that's probably because these large mesopredators were outcompeting the smaller ones and gaining access to the bait bag, which is pretty cool. But I think the most interesting result is the delay to feed in rays. So we saw a small delay to feed in the presence of reef sharks, so things like grey reef sharks and black tips, but a really big delay to feed in the presence of apex sharks, so things like tiger sharks and great hammerheads. And I think it's because when a shark arrived in the field of view, fish could just swim away because they're quite mobile. But rays actually rely on crypsis and their sting to avoid predators, so they're not as nimble, and it makes sense for them to exercise caution for quite a long period of time, especially in the presence of these apex sharks, which are often elasmobranch specialists and feed on rays and elasmobranchs quite a lot, like this great hammerhead that Molly saw feeding on a wedgefish up north. Okay, so I was thinking for these mobile species like fishes, how likely is it that the presence of a shark is going to result in a behavioural change that will last an entire hour? Well, when you think about it, it's probably quite small given the mobility of these species, and predation is probably quite an acute force that's only visible over shorter timeframes. So can the presence of a shark alter the behaviour of mesopredators over these shorter timescales? To do this, I looked at some more bruvs and I looked at the behaviour of mesopredators 5 minutes before and 5 minutes after a shark arrived in the field of view. And I looked at some different behaviours because those other behavioural metrics I developed couldn't really be applied to this sort of setting. And I looked at the amount of time that fish spent in the water column, because this is a very bold behavior. When fish are up in the water column, they're very conspicuous to predators. And I'm going to take, because this is a whirlwind tour, I'm just going to take you through the results of two fish, Lejanus boha and Lejanus gibbus, because they give you a nice overview of what we found. So for Lejanus boha, after sharks arrived in the field of view, they actually reduced the amount of time they spent in the water column. They pretty much ran for cover. And we also found that habitat was really important, was an important factor as well. So mean relief is a measure of habitat complexity, and the larger numbers just mean that it's a more complex habitat. And so at greater levels of habitat complexity, these boha were spending more time in the water column. They're being more bold, and we think that's because they have more places to hide compared to something like a sand flat where they're completely exposed. Lejanus gibbus also changed their behavior in response to the sharks, but it was only if the sharks were feeding on the bait bag. And the more bites that shark took from the bait bag, the less time they spent in the water column. Now that's probably because there's a very big difference behaviorally from a shark that's just cruising by the reef and a shark that's intent on feeding. It's a really obvious behavioral change. When sharks want to feed, they swim a lot faster. Their pectoral fins change from horizontal down to vertical. And that these gibbous were probably using these behavioral cues from the sharks to mediate their own responses so that they avoid exhibiting costly anti-predator behaviors to a shark that's just cruising by and doesn't represent a threat. So the next question is the question that Gary Kendrick always asks in student seminars. So what? What does this mean? What does it matter if a fish is spending less time in the water column? Well, it's actually probably quite a big deal, because fish that are down here, hiding under shelter and are on the substrate, can only access things, prey items that are down here too, things like invertebrates. Whereas fish that are acting a bit bolder and can go up into the water column can access different prey items like fish. So these changes in the behavior of fish mesopredators in the water column might be a way that reef sharks are behaviorally regulating mesopredators in coral reefs. And if you're interested in this, we just got it accepted in Ecosphere, so you can have a look at the details later. So let's bring it back to my original question. Can sharks alter the behavior of mesopredators in coral reef? Well, because of this PhD, I think we can say yes, and we can measure these changes using BRUVs, but it's a little bit complicated. Yes, these species do change their behavior in response to sharks, but the timescale of these reactions differ for different species. And these reactions are also mediated by factors of the environment, such as habitat complexity and the behavior of the shark. So this is a very important first step into understanding the ecological importance of sharks and coral reefs. But now we need to know whether these behaviors can cascade down to impact lower trophic levels, and what are the mechanisms and conditions that can facilitate this. And finally, we need to remember that we've examined these behaviors at Ningaloo, where shark populations are really healthy. But what we need to do now is to look at other places where sharks have been removed and to examine the magnitude of these anti-predator behaviors and to think about what impact this might have on an ecosystem. So just to summarize all of that, we found that reef sharks were in complex habitat that was and no-take zones are very important to this species. We developed some metrics to look at behavioral changes on BRUVs, and that we found that over long time frames, cryptic species increased their delay to feed in the presence of reef sharks and especially apex sharks. But for more mobile species, competition was a more important factor over these timescales. And that's probably because predation is an acute force, and which is observable over shorter time periods, but the responses were nuanced and context dependent. And finally, I just want to say a very big thank you. I can honestly say that my PhD was some of the best years of my life, apart from probably the last month or so before I submitted, even given that I got some wonderful, I did some wonderful things, and I did some experiments with model predators that I didn't even have time to touch on. They were really great. I went to Brazil to present my research at Sharks International and ASFB, and I even got to help out with some whale shark work while I was doing my stuff at Ningaloo. And so thank you so much for your support, and I hope you enjoyed my talk.
Thank you, Emily. And congratulations on your recently accepted paper and the other papers which will be coming. Thanks.
On their way.
Yeah. Our second presenter is Ankje Frouws, and she is the 2018 Keiran McNamara World Heritage PhD Top-Up Scholarship recipient. Ankje received an Erasmus Mundus Masters of Science in Marine Environment and Resources from the University of Basque Country in Spain in 2015, and she's currently completing a PhD at Edith Cowan University's Centre for Marine Ecosystems Research in the School of Science. Ankje's research topic is seagrass resilience patterns in genetic diversity of seagrasses in Ningaloo and Shark Bay. These seagrasses form critically important habitat and are vulnerable to disturbance. And Ankje is looking at the genetic diversity and connectivity of the seagrass populations and restoration efforts. Over to you.
Thank you very much, Tim. So in my PhD, I look at the resilience of seagrasses and specifically the patterns in genetic diversity of seagrasses in Ningaloo and Shark Bay World Heritage Sites. Why seagrass? Well, they're marine flowering plants with really important ecological roles. They provide habitat for fish and food sources for turtles and dugongs and other marine animals. They are able to store carbon in their soil and they stabilize sediment. So lots of reasons to care about them. And there are vast meadows in Shark Bay and in Ningaloo, but Shark Bay is a World Heritage Site because of their seagrass. And the meadows are vast, the largest seagrass meadows in the world, and you can even see them from satellite imagery. And you can see here on the left picture, these dark sort of lines in the water, that's all seagrass meadows. Majority of seagrasses in Shark Bay is the species Amphibius antarctica, the one you see here on the right. And it forms some sort of highway for fish when you dive through it. It's amazing to see. So unfortunately, even if you're a World Heritage Site, you're not immune to stresses. So there was a massive marine heat wave in 2010, 2011 that wiped out almost a quarter of the dense seagrass meadows in Shark Bay. And some of these meadows have recovered over time and some have not. And I look at the resilience of seagrass meadows. So the capacity of a meadow to either resist the disturbance, so deal with the disturbance and move on. Maybe a meadow will have a little bit lower density of seagrasses. Maybe there are small changes, but the meadow stays a meadow. Or the capacity of a meadow to recover. So a meadow might be wiped out, but then might be recovering. So a meadow turns into a sandy patch, but then becomes a meadow again. So those two processes, the resistance and recovery of disturbances by seagrass meadows, is considered the resilience of a seagrass meadow. So how does this resilience then relate to genetics? Well, if you look at seagrass meadows, on the two ones here in this illustration, on the left, you've got a meadow with different genetic information. So these DNA helices, the different colors represent different genetic individuals. So the meadow on the left here is consisting of different genetic individuals, while the meadow on the right is consisting of individuals which are all genetically similar. Well, that is fine if nothing happens to a meadow. But if some sort of disturbance occurs, the question is, how do these meadows respond to the disturbance? And if you have a lot of genetically different individuals, there's more likely to be a more diverse response to this disturbance as well. And you can imagine on the right, this meadow, if something happens to it, it might be completely wiped out, or it might be able to survive. But here on the left, the response is a bit more varied. Maybe some individuals don't survive a disturbance, but other ones are able to manage and deal with it. And in our marine systems, a disturbance often doesn't come by itself. You might have a heat wave. You might also have a change in light, for example, in a meadow, an influx of salt water or fresh water. So all these changes interact, and these interactions are quite complex. So one of the ways to ensure against such a complex interaction is to have a very diverse seagrass meadow. So I'm looking at that diversity in relation to resilience of meadows. Shark Bay is not only known for its seagrasses, but also for its really, really strong salinity gradient from north to south, especially in the eastern Gulf, where the salinity varies from sort of sea level salinity with 37 grams of salt per liter to over 50 grams of salt per liter, which is super, super salty. And because of this, what we know about Shark Bay is that it has vast seagrass meadows, but we don't actually know a lot about the genetic diversity of these meadows. So within my PhD, I look at that sort of the structure of diversity of seagrasses within Shark Bay. And I chose to sample along that salinity gradient to be able to capture as much diversity as I can. Because if there's any structuring force in that bay, it's very likely to be that extremely, extremely strong salinity gradient. So I sampled from north to south in the eastern and the western Gulf, in the yellow and the red dots. And then within these locations, took seagrasses, extracted DNA, sequenced it, and then look at the diversity, the genetic diversity of these meadows. I used a program called Structure to look at how similar these meadows are to each other. So I am in the data analysis phase, so a lot of my data is still preliminary, but here it is. So these are results from the Western Gulf. The dots that have a similar color are meadows that have a similarity in community. So lights are automatic here, that's great. So you see these green dots, which have a very similar community structure. And when there's a half green, it means that they share some community structure with these green dots, but the other ones is separate. And you can see that these sort of green genetic individuals are sort of spread from north to south. So there's some connection between them. And this site is very, very different. Now, if we take up the right meadows as well, you see the same sort of green individuals coming back in the populations, but then also some more diversity being added in. And it's quite interesting that you see more different populations on the eastern Gulf than on the western Gulf. One of the reasons could be is because these seagrasses in the eastern Gulf don't only provide habitat, but they also change the hydrodynamics of that system. So the seawater comes in from north to south, but then evaporates and doesn't really flow back. So the connectivity from a hydrodynamics perspective is very much more north to south than the other way around. So that could be one of the reasons. So what does this tell us? First of all, we didn't know that there was genetic diversity in amphibolous Antarctica in Shark Bay. Now we do. The next step for me is to identify how many genetically different individuals there are in each of these meadows and to calculate some more diversity statistics. And also just to study what is causing these differences between the different meadows. Is it a matter of connectivity or is it a matter of adaptation? Are there some locations where specific individuals are unable to survive and that creates these sort of different community compositions? And future studies can use this kind of data to look at, for example, what kind of individuals are more heat tolerant, which can be really useful for restoration efforts. If we look at this data in Shark Bay, it's just one moment in time. We looked at the diversity at one moment in time, but we don't actually know how much it changes over time. So I also looked at the temporal variability in diversity in seagrasses. Here on the left in the bottom is Shark Bay, and this map goes all the way to Bala Bala here in the northeast. And I resampled seagrass meadows of Halogile uninervis, a much more smaller seagrass species than amphibolous, with a more rapid turnover rate as well. And I resampled meadows that have been sampled 5 years ago. And I compared the diversity 5 years ago with the current diversity to see how variable that diversity is over time. And that results in sort of graphs like this, where you see the diversity in 2014 and 2019. And a diversity close to 0 means there is no diversity in the meadow. And a diversity close to 1 means that the meadow is really, really diverse. These data are clonal richness data. So that leads to a map that looks something like this. So remember again, the diversity of a 1 is very high and 0 is low. We see in general quite a stable diversity in that 5-year period. for several locations, and in other locations there is some increase, but it's not. It doesn't change a lot. We do see that the diversity in Shark Bay is much higher, for example, than the one in Ningaloo, making Ningaloo potentially more vulnerable. The meadows that are sampled there potentially much more vulnerable to disturbances than the one in Shark Bay. And the same for the Tavernard Island meadows, for example, while Exmouth has much higher diversity and could therefore potentially withstand pressures much more easily. So what does this all mean? Well, first of all, we see that there are small changes in diversity of Halogyli uninervis in that time space as well, but it's relatively stable over time, which indicates that if you use richness as a monitoring tool, looking at the clonal richness in monitoring, that could actually be quite useful because it stays stable over time. So the values you get today could still be used in a year, in a couple of years, and give you an idea of the resilience of that, the potential resilience of that meadow. We also see that these Halogyli uninervis species are quite fast growing. So we expect that diversity might even be changing slower in slower growing species with a slower turnover rate, like for example Amphibolus. So the next step in my PhD is to look at what is driving differences in diversity between the different sites. So comparing Ningaloo, comparing that with Shark Bay, comparing that with Bala Bala, but also why are some sites more stable in diversity over time than others? And bring that all together. So first of all, we can say, okay, yes, there is spatial variability in genetic diversity of Amphibolus antarctica in Shark Bay. We didn't know that before. There is also temporal stability in genetic diversity of Halogyli uninervis, looking at that sort of large spatial scale. But what does that mean for Shark Bay and for Ningaloo? Well, some meadows in Shark Bay are likely to have more resistant individuals than other meadows because of the diversity differences. And in general, populations are likely to persist for at least intermediate periods of time. So say at least a period of 5 years. Thank you.
Thank you, Ankje. Best wishes for the write-up of that work and also your next steps. It sounds exciting. Our next speaker is the 2019 scholarship recipient, Justin Geldard, who has a Master of Professional Engineering in Civil Engineering from UWA. He's a PhD candidate at UWA's Oceans Graduate School, and his research topic is hydrodynamic forces on coral reefs and improving the success of large-scale reef restoration, Ningaloo, colony to canopy scale. He's exploring how the structure of corals interacts with currents and storms, and using some novel 3D printing technologies. Over to you, Justin.
Thanks very much, Tim. I'll just share my screen. Thanks everyone for tuning in. As Tim said, my name is Justin Geldard and I come from a structural and coastal engineering background at UWA under the Oceans Graduate School, and my PhD is really focused on looking at hydrodynamic forces on coral reefs and how we can then apply this understanding to improving reef restoration. So just quickly an outline of the presentation. I'm going to start with a bit of background, move into the aims of my research, then touch on some preliminary results that we've just got and some future fieldwork upcoming and the impact of the research itself. So firstly, the background. As I mentioned, my research is in the Ningaloo World Heritage region, and I'm really focusing on this 260-kilometre long fringing reef system that you can see here. So of particular interest to me is the reef flat zone that you can quite clearly see in this image, and a common distribution of corals and species found on this reef flat is seen in this photo here. So this distribution of different species as well as density of actual individual colonies can be driven by different drivers. You can see on the left-hand side here, starting with light intensity, hydrodynamic stress, different sediment levels, as well as aerial exposure. But the real focus of my work is on the hydrodynamic stress component. So how does these hydrodynamic stresses change where we find corals across the reef? So firstly, what is hydrodynamics? So hydrodynamics is essentially just the movement of water, and wave hydrodynamics is then how water moves under waves. So you can see in this schematic here, we've got a surface wave traveling across the top there, and underneath the surface wave, we get these orbital velocities that have been drawn. Now these orbital velocities do attenuate with depth down to the seabed, which is where we would find our corals. But on these reef flats, the water depth can be quite shallow. So we get actually very high orbital velocities, which create an oscillating current across that reef flat. And you can see here in a shaky GoPro video, some of these waves traveling across the reef flat. And if you look at the small fish, you can actually see them slowly actually moving in circles. It's a bit of a tracer. Now, where do we apply the engineering part to this problem? So as a structural engineer, we look at an individual structure. So the coral you can see in the top left there. We then draw what we call a free body diagram of that structure, which is in purple there of the coral. And we draw the forces that act on that structure, which are in blue. So on the left-hand side there, due to the wave hydrodynamics, we get a drag force, which is due to the flow separating around that structure. And we also get an inertial force, which is due to the acceleration of the fluid through that structure. Now, these forces acting on the structure can either dislodge it from the seabed itself or break individual segments of that colony, obviously damaging the reef ecosystem and exposing further colonies within the canopy, which you can see in the schematic below. So when we move it to the canopy scale, flow can often be diverted above the canopy as the colonies work with each other essentially to shield more susceptible corals from damaging waves. And these are some of the consequences of these wave effects. So in this middle video here, you can see a branching coral that's lost some of its segments. On the video on the far left is a table coral that's been completely overturned. And then on the far right there, you see a landscape that we start moving towards, which is quite bare. As each individual coral gets damaged, it exposes other corals within that canopy to more damaging wave events. And there's also evidence of this on the shoreline of this beach here, which is all fragments of broken coral up in Exmouth. So the aims of my research are to significantly improve the understanding how the hydrodynamics associated with waves contributes to shaping the coral landscape, and then use this understanding to help restoration and management of where we should focus our efforts on in the Ningaloo World Heritage region. So the focus so far of my research has really been on the first aim here on the top left, on the forces acting on individual coral colonies. Soon we'll be then moving into what happens when we then introduce that colony into a canopy. So how do they work together to essentially protect each other from these damaging flows? Then thirdly, look how these canopies then protect our coastline from these damaging waves. So we often see wave breaking over reef flats, which then protects more fragile ecosystems like your seagrasses and your mangroves from these damaging waves. And then finally take all of this understanding into the field at Ningaloo and hopefully work with management and restoration teams to help, identify high risk regions and confirm this understanding. So quickly to touch on some of the preliminary results that we've obtained so far. So all of our work so far has been experimental in a wave flume that you can see operating on the top left there. So this is a 54-metre long new facility that UWA has just constructed. And on the left, we have a piston wave maker that moves backwards and forths and generates these surface waves, which then break on a beach down the other end. Now, along that wave flume, we have a 3D printed coral structure at the moment. And underneath that coral structure, we've instrumented a load cell so that we can measure the live reading of the force acting on this coral. And we've also got something called an ADV and a wave gauge, which measure the properties of the waves. So the first part is the 3D printing of these corals. So we obtained some skeletons from the West Australian Museum on the top left there. We then took that to the hospital through a CT scanner. And that CT scanner then gave us a digital model of the corals in gold. And then finally, we then take it to an industrial size 3D printer that you can see operating on the right, which can successfully recreate these corals quite accurately. And this obviously limits how much corals we're actually taking from the reef, and we can play with them in different sort of waves and adjust different parameters of those corals. So the current testing that we just completed, on the far left there, you can see on the base of the flume, we've just inserted a false floor essentially, and we've got a countersunk cup inside that, which is where the load cell sits. Then on top of that load cell, We have the coral that's mounted there, you can see in the third image. And some of the instrumentation coming down from the wood beam is your wave gauge measuring the height of the water surface, as well as an ADV, which measures your flow velocities that we would experience within the reef canopy. So just a quick snapshot of a time series of these results that we've been obtaining. So on the left vertical axis, you've got the horizontal flow velocity, and that's plotted in blue. And on the right-hand axis, you've got the force that's acting on different structures. So the solid orange line is the forces acting on a cylinder structure, which is quite commonly studied in hydrodynamic engineering. And the dotted line is the forces acting on our cylinder. So we can see here that they are slightly different. These two structures you can see on the right have the exact same volume, but they are slightly different in amplitude and also in phase. So we can see that the dashed line is becoming more closely in phase with the horizontal flow velocity, which is telling us something about the drag component, which is this bottom left component here, not as much as about this volume. So it's starting to reveal some information about how we can parameterize different corals based on their shape to how well or not so well they can withstand forces due to waves. So finally, just some future fieldwork and impact of the research. So when we head up to the Ningaloo region, we're going to take some of these corals that are instrumented and implant them within canopies within the Ningaloo reef, validate some of these findings, and hopefully work with different management and conservation teams to identify parts of the Ningaloo reef that are very vulnerable to hydrodynamic disturbances, potentially before the next cyclone comes through, so that we can implement some different restoration techniques in that area. Different restoration techniques commonly involve coral farming, which you can see on the right here. So coral farming involves often taking little fragments of coral, attaching them back to the reef, and then letting them regrow. Now, what isn't known really in coral restoration is which types of corals or which shapes of corals are best suited to different regions, and which ones will be able to survive in the wave climate if a cyclone came through, because the restoration effort can be wasted if it can't withstand those hydrodynamic forces. So we're hoping that our findings will be able to help both the management, conservation, as well as restoration in providing guidance on which species and which species distributions are best suited for those different regions of wave exposure. And that's all, and thank you very much for your support.
Thanks very much, Justin, and thanks for applying your mathematical knowledge to these important ecological questions, so thank you. Our final presenter is the 2020 recipient, Molly Moustaka. Molly has a Bachelor of Science with Honours from UWA, and she's just about to commence her PhD in the School of Biological Sciences here at UWA. She's been working as a research scientist at the Marine Science Program at DBCA. Molly's PhD topic is the influence of mangroves on diversity, abundance and productivity of marine systems of the Dampier Archipelago, included in the nomination for the proposed Murujuga World Heritage Area. Thank you, Molly.
Thanks very much, Tim, and thanks everyone for coming along tonight. As Tim said, I'm looking at the influence of mangroves on the productivity and diversity of the adjacent marine systems. In particular, we'll be looking at fish. I'd just like to thank the McNamara family and their committee for all of their support. It's going to facilitate some really interesting work, and also DBCA for facilitating the work that I'm going to do. So mangroves are structurally complex, highly productive plants that typically exist in sheltered marine bays. Their complex root structure provides shelter from predators and a lot of structure for additional food resources to grow on. So it makes them a really good nursery habitat for juvenile fish. They've been studied quite a lot in the Caribbean, which is a microtidal, permanently inundated system. So the fish can get in there all the time, and they've been identified as really valuable nursery habitats. In the Indo-Pacific, we tend to have more macrotidal systems, and the results of those studies are a lot more variable. So in areas with large tidal ranges, the mangroves are only inundated for part of each day, meaning that they're typically not the focal nursery habitat for fish. But what we're wondering is that given the benefits that they provide elsewhere, is it possible that they can enhance the nursery function of these adjacent habitats through the provision of periodically available shelter, foraging grounds, and also detrital subsidies, which can be consumed as food or potentially even fertilise these adjacent habitats. So really briefly on the life cycle of a lot of coral reef fish, it's multiphasic. So they start out with this pelagic open water phase and gradually move into these nearshore back reef habitats, which are made up of things like mangroves, seagrass meadows, macroalgal beds and shallow reefs, and they're typically sort of behind a fringing coral reef. And then gradually these species will move through multiple habitats back out to the coral reef throughout their life. So this is the phase, the nearshore habitats are the phase that we're focusing on. Given that the juvenile life history stage is a critical population bottleneck, the amount of successful recruitment to adult populations can be strongly influenced by food availability and habitat quality in this stage. And as a result, these back reef seascapes are being increasingly recognised as a priority for conservation and management because they're at risk of degradation from coastal development and also climatic disturbances, and can have disproportionate effects on the adult populations down the track. So when we say nursery habitat, what do we mean? Not all back reef habitat is nursery habitat. One of the most commonly used definitions of nursery habitat is from Beck et al., and it states that for a habitat to be a nursery, it needs to have four things: greater density of recruits, faster growth rates, greater survival of recruits, and greater successful movement to adult populations than other habitats. Recently, this concept has evolved to encompass the movement of fish over daily, tidal, seasonal, and ontogenetic, so life history stage scales. And this is called the seascape nursery concept. This concept says that the nursery habitat includes not only the focal habitat that the fish spend most of their time in, but also the habitats, any additional habitats they use for foraging, for shelter, or that provide additional subsidies, or that they use as stepping stones throughout their life. So while mangroves in northwestern Australia might not be this primary focal habitat in these macrotidal systems, we want to look at whether they can still enhance the functioning of the adjacent habitats by providing these periodically available resources. So where are we working? Beautiful Dampier Archipelago, otherwise known as Murujuga, including the Barat Peninsula, which means hip bone sticking out. It's an area of amazing Aboriginal cultural value and has been nominated for World Heritage listing just recently. So fingers crossed that that does come to fruition, because it's an incredible area. There's 42 islands, coral reefs, seagrass meadows, macroalgae beds and mangroves. It's a hugely macrotidal system, and it's also home to one of the world's largest bulk export ports. So the significance of this work: to date, the nearshore communities of Murujuga have been relatively poorly studied. This project's going to provide a better ecological basis for future planning and management and impact assessments in this potential World Heritage Area. It's going to further our understanding of the influence on mangroves and the abundance, diversity and productivity of adjacent producers and consumers, and will also further our understanding of carbon flow between nearshore systems, the role of detrital subsidies, and the links between primary and secondary productivity. So really quickly, my 4 chapters are probably going to be, at this point, it's quite early days, looking at in bays with and without mangroves, and in both coral and macroalgal habitat. We're going to look at the differences in habitat and fish assemblages, and to try and examine whether mangroves are having a measurable effect on total fish assemblages. Then we'll look at recruits specifically and relate that to the habitat variables and also to some environmental variables, and clarifying the importance of connectivity to mangroves. Thirdly, we're looking at differences in growth rates, which is one of those key metrics of a nursery habitat, and then relating that to habitat quality, environmental variables and mangroves. And lastly, we're going to use stable isotopes and gut content analysis to look at the diets of juvenile fish, looking at the relative trophic input of mangroves, and combined with the other chapters will let us understand the degree to which mangroves are supporting nearby marine communities and the cause-effect pathways through which this is occurring. Okay, thank you for that sprint. That's me. Thank you very much.
Thank you, Molly. Thanks for that work on the mangroves and connecting the marine world to the terrestrial world. So that's our presentation. So thank you to all of our presenters for your fascinating talks and various disciplines. It's very exciting to see all the different approaches and areas being studied. I'd like to now open up for questions from our presenting panel. Would anyone like to ask any questions?
Maybe I'll start there, Tim, if you don't mind.
Yes, thanks, Glenn.
Firstly, I'd like to say fantastic talks by all of you. They're really, really good. Really good to see some really nice science come out of this program. So, and obviously you've been working in some really nice and fun environments. So fantastic. My question was to Emily. So nice talk, Emily. Really enjoyed it. Really, really clear, and some nice outcomes from your PhD by the sound of it. I was just wondering, in terms of the brubs, what sort of influence do the brubs themselves have on the behaviour of fish, and how might that actually influence some of your observations in terms of their response to sharks and other major predators?
Thanks, Glenn. That's a really good question. Yes, the answer to that is, brubs are attracting things to the bait. So that is going to have an influence on the behavior of the fish. But that's sort of what we're interested in. We were sort of interested in the trade-off between predator avoidance and accessing the resources. So it's sort of the aim of the experiments, to be honest. And also, I don't know how you would go about recording these sort of interactions without bait, given the mobility of these species. So unfortunately, it's just what we had to do to get them in the same place.
In terms of the structure of the reefs and so on, and I wasn't quite sure how close those brubs were and whether they were all sort of pointing towards the actual reef structure or not. So it'd be pretty handy to have some of that information.
Yes, so obviously we're only limited by what we can see in the field of view, and one of the limitations of brubs is you can look out and see a sand flat, but perhaps right behind the brub there's some wonderful complex coral reef habitat, and it's just something we have to deal with. A 360 brub would be ideal, so you could get more of a panoramic view, but yeah, that's a good point too.
Nice.
Thank you.
Yeah, I have a question for Ankje. Very interesting talk, Ankje, and I know it's really early times with your data and really interesting patterns. Have you seen anything that sort of stands out as potentially why some of those seagrass meadows are surviving where they are in relation to that 2011 heat wave? So is it areas with high diversity or not?
So I expect a question from you, Gary. I would be disappointed if you wouldn't have asked. Thank you. So the locations where I sampled were specifically selected to not have undergone any damage during the heat wave, to make sure that we get sort of a more long-term history within those genetic data sets as well. So I did not sample any locations that were affected. I specifically avoided those. So that is a little bit difficult to answer the question in that regard. And also, when you look at sort of where the most damage was, it was mostly in sort of deeper regions within Shark Bay. And my sample sites are also more or less on the same depth, which is a maximum of maybe 3 meters, more or less, but most are shallower as well. So I would say, to answer that question, we probably need to sample some deeper sites as well to compare. I am really interested in seeing if I can find any, so the next step for me is to identify any loci under selection. So that would be able potentially to answer more questions, but possibly more in sort of adaptation to salinity, more likely than any heat wave related. But follow-up research, definitely.
Yeah, one of the ways to tackle that would be an experiment. So actually, your inference is increased diversity equals increased resilience. Might be really interesting to tackle some sort of large-scale lab experiment, or even an experiment in the field to test that. Have you thought about doing that?
It is on the wish list. Time is limited as well.
I'm smiling a lot because that's a lot of work.
It is a huge effort, yes. So one of the things we're considering at the moment is to do some sort of exchange of genetic material between different locations within the bay to see if specific genetic individuals are able to survive in other locations as well, or if they would be better off where we found them. So that kind of exchanges answers some questions, but it's not necessarily the same as exposing different genetic individuals to different temperatures and then seeing how they survive. I think that would be beyond the scope, but some sort of transplant experiment would potentially still be possible.
Well, if you get there, please invite me along. I would love to come and give you a hand.
I will, Gary, no worries.
Hi, I've got a question for Molly. So I was interested in your fish gut stable isotope and item analysis. My question is, how would you quantify the amount of biological matter that's moved by fish from mangroves to the coastal regions adjacent to them?
So I've just put this as a caveat that my first day enrolled was today. So I'm actually very, very early in the process.
No, that's fine.
I think more what we're going to try and do is collect the recruits and understand the proportion of their diet that's directly stemming from mangrove carbon, or other primary producers. I know of other studies that have gone and actually sampled the adult fish on the offshore, or not necessarily the offshore reefs, but the outer reefs. And you can actually take the otolith, the ear bone, out of the fish, the adult fish, and drill out the core and do stable isotopes, compound specific stable isotopes on that, and quantify the amount of mangrove carbon they were consuming as a juvenile, and then look at their growth rates with that. I would love to do something like that. At the moment, it's insanely expensive. You know, it's one for the wish list. If I can make it happen, I will. I've had a few conversations with some people that are doing that. That's sort of the last step of that process, is actually trying to understand how many of those recruits are successfully making it out to the reefs, and that's probably one of the hardest pieces to actually quantify. So fingers crossed I get there in the future.
Sounds great.
I have a question for Emily. You had those models. Was that a successful approach where you had the fake animals and did experiments in the field with them?
They were actually surprisingly successful. They really did fool the fish. We had remoras trying to attach to them, and I had some models of non-threatening species too, and a shark actually tried to eat my coral trout model, so they worked really, really well. But we found that they worked over shorter time periods. So I was looking at, again, feeding behaviour in response to the models of sharks. But once the fish started feeding on the bait, they sort of realised that the shark wasn't responding in the way that a real shark should. So they kind of just went for it after that. So yes, they were good for looking at these immediate predator-prey interactions, and they were really fun to work with in the field, but they weren't so good for these longer-term interactions.
Thank you. And if I'm allowed, I've got a question for Justin as well. Justin, when you instrument up those model corals, and so the flume is just fantastic to have as a resource to challenge the corals and measure everything. But when you take that to the field, you know, how robust is it in terms of being able to measure the things you want to measure, or are there some major technical challenges in logging the data when you're in the field?
Yeah, it's a good question. So that's something we're working on at the moment. So we're aiming to not have to be out there while logging it, the data itself, because some of the instrumentation that we use in the flume is quite fragile because it's measuring very high frequency and high accuracy. So when we go to the field, we do have to compromise a little bit of that, but we're aiming to have a setup that can be left in the reef by itself, and we can come back in a week or two and collect that data. So yeah, we're working on a casing or a housing at the moment that we can attach securely.
Okay, good luck, because that'll be such an advance. Glenn.
Yeah, while Justin's having a bit of a talk there, just found your talk really interesting, and it's nice to see some sort of physics and stuff come into the area. How, in terms of the flume equipment that you've got, how extreme can you create the force conditions? So can you bring those forces up to something like a cyclone event that you might get on a reef in extreme conditions?
Yeah, so I mean, there's two ways to look at it, in that it's a scale question, or we can scale things appropriately, so we can mimic real conditions from the Ningaloo into that wave flume. The maximum wave height we can generate in there is 0.5 metre, which is quite large. In relation to cyclone conditions, we can use another facility which is actually in the same warehouse, called the O-Tube, which is just essentially a ring that has the same thing, no waves. It's just highly accelerated flow. So there's an impeller that generates long and very high flows up to 4 meters a second. So there's multiple ways to test these conditions. The flume's good at those really small conditions as well, because we can drop that water depth really low and get some aerial exposure.
Yeah, because I would have thought that the cyclone conditions are really going to be the type of conditions is going to have the most impact on those, on a whole range of different coral species, and of course that's going to have a huge implication on the recovery programs and the reseeding programs. So getting some handle on those sort of more extreme events would be fantastic.
Yeah, that's exactly right.
Tim?
Sorry, Margaret, yes.
Yes, I was just going to ask Ankje about the seagrass dynamics and the genetic diversity, and have you done anything in terms of relating that to the spatial water flow dynamics around all of those areas and how connected the populations are or aren't?
Not really. So it's more almost the other way around. So looking at connectivity and then inferring that there is probably some genetic exchange. I'm not looking at the currents as such. I am using some model data from UWA, or sort of that's happening in the future, to look at, for example, the exposure of different cells within Shark Bay to different environmental conditions. So what is the depth? What is the salinity there? What sort of temperature exposure is there sort of on average? So those kind of things, but not really planning to look at hydrodynamics just because it's a whole different ballgame, or it's quite a large project. So it's not on the schedule at the moment, no.
Okay, thank you. So I've just received a message that we're near the end of our time together. So thank you everyone for joining us. Thanks for attending. I'd like to thank our 4 speakers for their fantastic talks, showing the diversity of studies that are happening and have been enabled by this scholarship. So it's important work carried out by 4 impressive candidates, and this wouldn't be possible without the Keiran McNamara World Heritage PhD Top-Up Scholarship. So we really appreciate the support from the McNamara family, and we look forward to the ongoing work by our current candidates. And Emily, we wish you all the best for your next step in your career now that you've finished. So thank you everybody, and we look forward to catching up with you all again in the near future, and best wishes to you all. Thank you.
Thanks, Tim, and thank you to everybody for presenting. It was really excellent. Thank you.
Thanks, Margaret.